Great. Good morning. The plenary talks so far have been extraordinarily enlightening about all the different facets of network science, the methodologies, the different systems, the different slices of reality that are being studied by various network scientists in various disciplines. In the next talk, I think we'll flesh out this picture further. Very pleased to introduce Professor Alex Fornito, who is joining us from Monash University in Melbourne, Australia. He received his PhD at the University of Melbourne in psychology, went on to do a postdoc at the University of Cambridge, and is currently an associate professor at the Monash Institute of Cognitive and Clinical Neuroscience. He has contributed a great deal to the emerging field of brain network analysis and network neuroscience through many high-impact publications and really seminal studies that have opened our eyes to the ways in which brain networks change through time, the way they are tethered to genetic and molecular underpinnings. He's the co-author of a extraordinarily useful textbook on brain network analysis published last year by Elsevier. And I think he will tell us a lot about how networks in brain systems are connected to each other, how they evolve through time, how structure and function inform each other, and how the functioning of brain networks is supported by the underlying molecular substrates of cells and tissues. So without further ado, please welcome Professor Alex Fornito.
Well, thanks very much for that kind introduction, Olaf, and thanks for the invitation to come and speak here. So today, as Olaf mentioned, I'll talk about some of the work that we've been doing trying to bridge different scales of resolution in the analysis of brain networks, and really, I guess, trying to unpack the molecular basis of the large-scale topological organization of brain networks. And really, this work is geared towards understanding what many come to view as the two fundamental principles of brain function, and that's segregation and integration. So on the one hand, we know that specific parts of the brain are specialized to perform particular functions. And this idea can be traced back to Franz Joseph Gall, working in the 1700s. He thought that you could go and measure the various dents and lumps on someone's skull because these were driven by the size of the brain region underneath that area. And the size of that brain region was driven by the degree to which a particular character trait was prominent in someone's personality. So he did that with people from all walks of life and generated a map that looked like this. And it's a bit fanciful. He's got traits such as destructiveness over the ear and language under the eye and memory in the forehead. And so we now view this as pseudoscience, but the basic idea that specific parts of the brain are specialized to perform particular functions has been a powerful organizing concept in neuroscience since then. And so if we fast forward to the middle of last century, we had maps like this doing the rounds. And this was really a synthesis of a large number of lesion and electrophysiological studies indicating that if you focally perturb a particular brain region, you can often elicit quite specific behavioral effects. Now with the advent of modern brain imaging techniques, we've become increasingly sophisticated in our ability to generate these maps, and a nice example was provided a couple of years ago with this article published in Nature, where they mapped the brain regions involved in processing the meaning of individual words from natural speech. So our understanding of functional specialization and segregation in the brain is quite advanced, whereas our understanding of how these segregated functions come to be integrated into the coherent experience of the world that we all enjoy has lagged behind. So this is a map of the macaque visual system published in 1991 by David Van Assen and Dan Fallerman. And it was really the first attempt to comprehensively map the connections between a large-scale brain network. And the year after, Francis Crick of DNA fame, together with Edward Jones, published a famous essay in Nature where they congratulated the authors on that attempt, but really bemoaned the fact that we had no methods available for generating comparable maps in the human brain, despite the importance of connectivity and integrated processes. And it was an auspicious time for them to be writing this because a year earlier we had the discovery of the Bold effect, which is really the basis for most of the functional brain imaging that we do today. And diffusion MRI had been around for a while, but it was really a few years later where we saw the first tractography studies. So these are studies that used a particular form of MRI to try to reconstruct the axonal connections in the brain. So by the time we got to the new millennium, our techniques were sufficiently advanced that we were able to map brain connectivity in living humans. And so this led Olaf Sporns, Giulio Tononi, and Rolf Cotter in 2005 to call for a map of the human connectome, which they defined as a comprehensive structural description of the network of elements and connections forming the human brain. So notice here that the emphasis is on a network description. And really, when we're talking about the brain, we can identify networks at all different scales of resolution. So at the nanoscale, we can define networks of molecular interactions that drive synaptic signaling. At the microscale, we can build networks of individual neurons connected to synapses. At the mesoscale, we can map axonal projections connecting populations of neurons. And at the macro scales that we typically look at in analyses of human data, we can map large white matter fiber bundles between broad brain areas. And so I'm going to begin by talking a bit about our work focusing on the macro scale in humans. And there's a fairly well-established pipeline for building connectomes in human brain imaging data, and listening to Jennifer's talk, I will forever be grateful for the relative ease with which we can acquire our data. But the basic idea is that we parcellate the brain into distinct regions, and they act as our network nodes. And then we measure some aspect of structural connectivity or functional coupling in the activity profiles of those areas. And from that, we build an adjacency matrix and a graph representation of the network. And then we can do any number of different topological analyses, which I'm sure you're all familiar with. And so in our own work, we've focused on understanding interactions between two large-scale and fairly well-characterized networks. So in orange, you can see what's variably called the executive network or frontoparietal control system. And it typically shows increased activity when people are performing a very difficult, challenging task. And in blue, you see what's called the default mode network. And it typically shows reduced activity during these tasks and increased activity when you're attending to your own internal thoughts. And so it's long been thought that these two networks have a competitive or antagonistic interaction. So when one is active, the other is deactive. And certainly if we look at complex cognitive tasks, we do find evidence for strong segregation. So this is just a force-directed plot of the nodes in each of those networks. And you can see quite a strong segregation between them. And this was characteristic of people who performed well on the task, whereas poor performers showed less segregation. But then if we look at other task contexts, such as memory tasks that require people to attend both to external and internal stimuli, then we find that greater coupling between these systems supports better performance. And this is facilitated by a shift in the modular organization of the networks. So this balance between segregation and integration is quite dynamic and context dependent. And if we just look at how network dynamics evolve spontaneously through time, so we put people in the scanner, we don't get them to perform any particular task, we also find that there is this spontaneous alternation between integration and segregation. So here I've plotted the mean regional efficiency of every brain region. And so this is really a measure that's inversely related to the path length. So it's a coarse measure of how integrated each region is with the rest of the network. Red is high integration, yellow is low. And you can see that there are periods where almost the entire brain goes red, followed by periods where almost the entire brain is yellow. So corresponding to these high integration and low integration states. So suggesting that the brain is spontaneously toggling between these segregated and integrated topological patterns. So what kind of structural network can support this dynamic balance between segregation and integration? Now, a lot of work has gone into understanding this, and I'm going to try and summarize it with a single slide. But the basic idea is that the brain can be decomposed into modules, so represented by different colors here. So these are subsets of nodes that are densely connected with each other, and it's thought that they serve some kind of common function, so they represent a specific functional system of the brain. And each of these nodes has one or more hubs, and these hubs are densely interconnected with each other, forming what we will call a rich club. And so on the one hand, we have functional segregation and specialization supported by modular architecture. And on the other hand, we have this hub connectivity and rich club organization that supports integration between the modules. So you can imagine that if I want to send a message from this module to this module here, I need to go through this information superhighway of hub connectivity to do so. And so I've also allowed for overlapping modular organizations, so a lot of these nodes belong to multiple modules. Now, the interesting thing about these hub connections is that they're topologically central, so they play an important role in network function, but they're also very costly. So generally, they extend over longer anatomical distances than other connections. So they occupy greater volume within the limited real estate of the skull. They require greater cellular material and consume more metabolic resources. We can see this with metabolic imaging. So this is data from Marcus Reichel's team, where at the top they've mapped the areas showing high levels of energy consumption. And you can see that spatially they correspond quite nicely with key nodes of the default mode network and the executive control system. These are really key areas of association cortex that are the hubs of the brain. Now, the interesting thing is that these areas that are quite metabolically demanding seem to be particularly vulnerable to the effects of disease. And you can see this with this image here, which shows amyloid accumulation in people with Alzheimer's disease. You see there's quite a strong spatial correspondence between the areas showing high levels of amyloid deposition and the metabolically hungry hubs of the brain. And this idea was recently expanded by Ed Bullmore's team in a meta-analysis of over 26 different disorders, where they found that reductions in grey matter volume tended to occur in areas that were high in degree. So it seems that across different disorders, regardless of the underlying pathology, dysfunction and abnormalities tend to accumulate within the hubs of the human connectome. So on the one hand, hubs are functionally valuable, they support integration, and on the other hand, they're costly and perhaps vulnerable to dysfunction and disease. And so this trade-off between cost and value is interesting to us because a few years ago, we did a small-scale twin study that found that a metric that tried to capture this balance between cost and efficiency was under quite strong genetic influence. So up to 60 to 80% of the variance in this measure between people was explained by genetic effects, and it was specific to this measure that captured the balance between cost and efficiency. We didn't see similar genetic effects for just cost or efficiency alone. So it suggests that evolution may favor connections that provide functional value, support integration for relatively low connection cost. Now, that's some analysis at the macro scale, but we wanted to unpack the molecular basis of this genetic effect in a little bit more detail. And to do that, we've turned to analyses of the mouse brain. And so this is data acquired at a mesoscale, so looking at axonal bundles between populations of neurons. And we link it to molecular processes, so at the nanoscale up there. And so we've used data provided by the Allen Brain Institute. A couple of years ago, they published their first draft of the mouse connectome from a large series of track tracing experiments. So the basic idea is that you inject a virus into a specific part of the brain, and over the course of a few weeks, that virus propagates throughout the connections of that areas and gets taken up in connected sites. So then later on, you can image where in the brain that tracer has been taken up. And so here you can see on the left, the top left, these are all the injection sites. They did over 400 different injections into the right hemisphere. And these are examples of the images you get mapping the connections for a given connection site. And so then they combine all of these data into a single connectivity matrix. So we end up with full connectivity for all right hemisphere regions to other right hemisphere regions. And we've also got the projections running from right to left. But I'll only talk the right hemisphere network today. So our first step was to just look at the centrality and cost of hub connectivity. And here what I've plotted is at the top you can see the degree distribution, so you can see an extended tail consistent with the presence of hubs. And in the first case we just looked at whether the mouse brain shows rich club organization. So this is something that's been seen in the worm nervous system, in the human, in the macaque, the rat, and here we see evidence for it in the mouse. So this is a normalized rich club coefficient. So along the x-axis we have degree, and at each level of degree we look at all nodes with degree greater than that number, so degree greater than k, and we say what's the density of connections between nodes above a certain level of degree, and we normalize that with respect to an ensemble of degree matched random networks. And so values greater than one suggest there's greater connectivity between hub regions than you would expect by chance. And so that's what we see as we increase degree, you see an increase in this rich club coefficient until we get into the tail of the distribution here, and this grey shaded area is what we call the topological rich club regime. So hubs of the mouse connectome are more densely connected than you would expect by chance. This image just shows the anatomical distribution of the hubs. So each color represents a different anatomical division, so cortex, cerebellum, thalamus, and so on. And you can see that we've got hubs present right across all the different anatomical divisions. So it's not confined to any single area of the mouse brain. And here I've got three different measures of connection costs. So we've got the mean connection distance, the probability of a reciprocal connection, and the average connection weight, which is related to the number of axons between two areas. And in each case, you can see that they increase as a function of degree. So again, indicating that the connections between hubs are the costliest elements of the mouse connectome. And then here we've got two different measures of centrality. We've got the mean between the centrality and the mean communicability. So they just make different assumptions about how dynamics unfold on the network. And in both cases, you see an increase as a function of degree. So again, showing, consistent with a lot of other work, that hub connections are both central and costly in the mouse brain. Now, the nice thing about this data is that for each of our brain regions, we have measures of gene expression in over 17,500 genes. So on the left here, I've got the connectivity matrix, and on the right, we've got a gene expression matrix. Each row is a different brain region, and each column is the expression value in one of over 17,500 genes. And so we wanted to try and relate these two in some way and ask, well, do hub connections show a distinct genomic signature? And so to do that, we split the brain into hubs and non-hubs. So hubs are shown in orange in that schematic and non-hubs in blue. And once we do that, we have three types of connections that can occur. We have what are called rich links, which are a connection between two hubs. Feeder links, which is a connection between a hub and a non-hub. And peripheral links, which is a connection between two non-hubs. And this is following a designation first proposed by Olaf Sporns and Martin van den Heuvel. And then for any given region, we can go into our expression matrix and pull out the corresponding vector of expression values, and then we can compute a correlation between any pair of expression vectors. So then for every pair of brain regions, we've got the type of connection that they're linked by, and a measure of how correlated their gene expression is across the genome. Oops, keeps jumping, sorry. And in the first instance, we just ask, well, does this gene co-expression value vary as a function of the type of connection a given pair of regions is linked by? And so here, in red, I've plotted the co-expression for rich links, and then green feeder and blue peripheral. And you can see that co-expression tends to be highest for rich links, and there's a sharp increase that coincides with the onset of the topological rich club regime, intermediate for feeder, and not significant for peripheral links. So connected pairs of hubs show higher coupling of gene expression than other pairs of brain regions. This slide just shows the anatomical distribution of the findings. The details are not so important. It's just consistent with the other image I showed, suggesting that the effect is distributed throughout the brain, not confined to any single anatomical division. Now, this effect is quite broad. We're talking about a correlation across 17,500 genes. So then we developed a way to determine the contribution that individual genes make to this overall coupling. And we fed it into an enrichment analysis. So we're asking, do certain sets of genes make a differential contribution to this effect? And we found evidence for enrichment in 5 different gene ontology categories that really fell into 2 non-overlapping clusters. So these were genes involved in regulating the synthesis and metabolism of ATP. So ATP being the primary energetic substrate of neuronal activity. And if we just plot the co-expression values for these 5 categories, you can see that there's a really strikingly specific molecular signature that coincides almost precisely with the onset and offset of this topological rich club regime. So indicating that hub regions have got more correlated gene expression, and this effect is driven largely by genes regulating energy metabolism. Now this signature was specific to hub connections. So if we just compare connected to unconnected regions, ignoring their topological properties, we see enrichment for all of these different categories, and there's a lot of things that you'd expect to see. So things like regulation of synaptic plasticity, neuron-to-neuron synaptic transmission, axon elongation, and so on. So these data are telling us that while these genes are important in establishing a connection in the brain, the primary genomic distinction between different topological classes of connections is really driven by the metabolic demands of the regions that those connections are linking up. So that was identifying a genomic signature for hubs. We were next interested in, well, do hubs show a distinct dynamical signature? And so to do this, we started with our anatomical connectome, but then looked at some fMRI data acquired in anesthetized mice in collaboration with Valerio Zerbi and Nicole Venderoff in Switzerland. So for every brain region, we had a time series reflecting intrinsic neuronal activity. And obviously, there's a range of different measures you could compute from those time series. And rather than focus on any specific ones, we took advantage of an analytic framework that a postdoc in my team, Ben Fulcher, developed. And so this allows the estimation of some 7,000 different univariate time series properties. So varying from basic measures such as standard deviation and entropy to autocorrelation and various model fits and so on. So then for every brain region, we have 14 different topological measures describing its structural connectivity. So these are different measures of centrality and clustering. And then we have just under 7,000 different properties describing the time series, so the intrinsic dynamics of that region. And so then we asked, well, how do these time series features correlate with structural topology? And we found that the most robust correlations were observed specifically for the weighted in degree of a region. So we lose a lot of information if we binarize the network and symmetrize it. So specifically the weighted in degree. And the measures that correlated with weighted in degree really clustered into one large cluster that was related to the spectral content of the signal. And so this is just an example scatter plot for one of those measures, which is the high frequency power on the y-axis. You can see there's a negative correlation with weighted in degree. So essentially areas with a high weighted in degree, a large number of strong incoming connections have got relatively higher power in low frequency bands. This is consistent with recent modeling work from Xiaojing Wang's team, where they took the macaque connectome, 29 different regions, and they simulated a biophysical model of dynamics on that network, and they looked at the effects of an input to visual areas, and they mapped the responses of different brain regions through the cortical hierarchy, so from low order sensory areas through to higher order association regions. And they found that as you move up through the hierarchy, you get a slower time scale of responses. So you can see this is V1, quite a rapid response that corresponds to the onset and offset of the input. Whereas as we go up in the hierarchy, the time scale of the response is much slower. And they found that this effect was really driven by regions that have long-range projections. So areas that have got these costly connections that integrate different areas, they've got these slower dynamics, and they found that this time constant of activity was really a strong predictor of the effect of a given brain region on overall network dynamics. So if you pull each region out and then look at the change in functional connectivity of the remaining areas, it's almost perfectly predicted by the time constant of the region's activity. So together these findings are suggesting that hub regions of the brain, particularly those areas with a lot of incoming connections, are characterized by a slower timescale of activity, which potentially allows them to integrate a diverser range of inputs. So there were some initial characterizations linking molecular processes to the mesoscale organization of networks. And then the next question is, well, would we see similar properties if we changed scales? And so to address this question, we've gone to the microscale and we've focused on analyzing the nematode worm C. elegans. This is quite an interesting organism. It's got a very stereotypical nervous system, 302 neurons, and it's the only organism to have had its connectome mapped almost entirely at the level of each and every neuron and synapse with electron microscopy. So the data are as real as they get. And we also have gene expression measures available for the worm. Using the open worm-based dataset, it's not as complete as in the mouse, so it's based on the curation of published findings, and it's binary data, so we only know if a gene is expressed in a neuron or not. So on the left here, I've got the connectivity matrix. In the middle, we've got the gene expression matrix, so each row is a neuron and each column is a gene. And then on the right, we can compute a gene co-expression matrix, a neuron by neuron co-expression matrix, just by estimating a measure of correlation between binary vectors. And we know from work from Ed Bullmore's team that the worm shows rich club organization. And so here I've just shown where the hubs are in the worm. They tend to be in the head and the tail. And you can see that these rich links extend almost across the entire body of the worm. So again, they're quite costly. And the hubs also tend to be interneurons, as opposed to sensory or motor neurons or multimodal neurons. And so then in the first instance, we asked, well, can we replicate this effect? Do we see enhanced gene co-expression in connected pairs of hubs? And indeed we do. So as you can see here, in red, you can see that the rich links, so the connections between hubs, have got on average higher co-expression than other types of links. And this increases as a function of degree. And the nice thing about the worm is that we know a lot about the individual properties of neurons. So we can ask, well, is there anything specific that's driving this effect? And as I mentioned, most of the hubs are interneurons and they're located in the head and the tail. So is this effect driving it? Well, not really. So for example, if we look at just interneurons and we compare hubs to non-hubs, we find elevated co-expression just in the hubs. We can then do community detection, and we can ask, well, does the modular organization drive this co-expression finding in any way? So in this case, we find evidence for 4 communities, and most of the hubs are located within a single community, this one here. And if we just compare inter and intramodular links across the network, we see no difference. And if we focus just on this module here, and we look at the intramodular links of the hubs versus the non-hubs, we see an elevation just in the hubs. So modular organization in general doesn't affect gene co-expression, and there's nothing special about being in this module alone since the effect is specific to hubs. Now, the nice thing is that we know the birth times of each neuron in the worm, and again, Ed Bullmore's team has shown that hub neurons tend to be born quite early in development, but if we compare co-expression of hubs born early in development compared to non-hubs born at a similar time, we see that the co-expression is elevated just in the hubs. So it's not got to do anything with birth date. And then finally, we know the lineage distance for every pair of neurons. So this is the number of cell divisions that separate 2 neurons from the common progenitor cell. And if we look at the distribution of distances for hubs and non-hubs, again, there's a lot of overlap. So that can't explain the findings either. So to the best of our knowledge, it seems that there really is something specific about being a hub neuron in the worm nervous system that's driving this enhanced co-expression effect. And that occurs despite these neurons being separated by a longer anatomical distance than other types of neurons. So to summarize, brain networks are topologically configured to support a dynamic balance between segregation and integration. Modularity supports segregation and hub connectivity supports integration. Hub connectivity is a central yet costly aspect of network topology, and this is something we see across different scales, and it seems that the intrinsic dynamics of hub regions unfolds over longer time scales. hub connectivity has a distinct genomic signature. So in humans, we see that network cost efficiency is heritable, and we also see that hubs show enhanced coupling of gene expression, and this is driven by metabolic genes. And this genomic signature is preserved across different scales. So we see it in C. elegans in the mouse, and some recent data from Ed Bullmore's team using a very different analytic approach indicates that areas with high participation coefficient also show increased expression of metabolic genes in the human brain. So we're seeing it across lots of different scales. So I'd just like to close by thanking the people who did the work. So Ben Fulcher did the mouse work, Arena, Anna Kevichute did the work in the worm, and the human work was done together with Michael Breakspear and Ed Bullmore. As Olaf mentioned, with Ed and Andrew Zaleski, we published a book last year for anyone who's interested on network analysis in neuroscience. Some links to software and just a quick plug for Network Neuroscience, which is a new journal founded by Olaf Sporn. So please send your work there if you're in the field.
So in your macroscale data, is there evidence that there are neurons that are part of the hub structure, or could this all be sort of axonal superhighways? You used the word superhighways, which made me think about this question.
Not quite sure what you mean. So in the macroscale data, we can't resolve individual neurons. So we kind of pull data over a broad area which contains millions of neurons, and so really all we can map are the fibers that come in and out of those areas. And so we can say that two of these populations are connected or not and get a rough estimate of the strength of connectivity, but we can't resolve individual neurons.
Right, so when you refer to populations in that setting, we can't take for granted that those are actual populations of neurons, right? That area that's lighting up as a hub that's highly connected to other areas could actually consist of fibers of passage and long-range axons and so on, or would you argue against that?
So in, for example, the measures of brain activity, the signal is driven largely by neuronal activity. And the regions, the fibers that we get coming in and out of areas emanate from neurons. But really, it's only in the worm that we can resolve individual neurons.